Before we look at today's work, let's look back to last class.
We looked at traditional classification in biology, the classification system that's been around since the time of Carl Linnaeus, when he created Systema Naturae and he set up the system of binomial nomenclature, the two names for the organisms that we have, the genus name and the species name. And we pointed out then that biologists have agreed through a series of high-powered meetings or congresses that the binomial system will be maintained at least for the next 50 years. Species like gorilla, iguana, Drosophila melanogaster, Bufo marinus, all of those, Lumbricus terrestris, all of those that we've been studying, the genus name and the species name, they will be maintained as is. But we can't say the same for the hierarchy: domain, kingdom, phylum, class, order, family, genus, and species. And the reason that we can't say that it's going to stay has to do with new evidence that's come to light from DNA. DNA itself was only discovered and understood properly in the 1950s. Its structure and how it replicates, and then how it leads to protein synthesis. Of course, the power to read these DNA sequences, or what we now know today as gene sequencing, protein sequencing, the whole field that's come about because of recent advances in biology over the last 50 years or so. This entire field of molecular cladistics has led us to revisit the way that Linnaeus laid things out and to look at things in a new way. So this is why in section 5.4 we are going to look at the science of cladistics, but we're also going to connect it to another part of the IB Biology Guide, which is section 3.1, and I'm going to point out the details of all of that for the next couple of lessons. But for today, I want to go into section 5.4 of the guide and to point out that a clade is a group of organisms that have evolved from a common ancestor. And cladograms are tree-like diagrams that show the most probable sequence of divergence of clades. So what's the big deal with all that, you might ask? We've got the mosses that we studied, we've got the ferns that we studied, we've got the conifers that we've studied, and we've got the flowering plants. The fossil record will show that mosses were around a long time ago. When life first emerged out of the oceans, the first plants to colonize the banks of land were the mosses. Then, in time, ferns began to appear on the fossil record, and then conifers, and finally the fabulous flowering plants that are so ubiquitous and we find all around us today. When these four groups are laid out in a sequence like this, with an algal ancestor down here and each of the groups diverging in time, I've highlighted the significant differences as we go from mosses to ferns, then to conifers, and finally to the flowering plants. All of this is a cladogram. Why do we need a cladogram when we've already got everything laid out like this? We've already identified that the plant kingdom is divided into these various groups. The reason is, this particular cladogram has no problem with it. And most cladograms that we are going to construct would have no problems. We can look at gene sequencing, we can look at protein sequencing, and it would prove what we knew all along, what we have fossil evidence to show, and evidence from looking at these things, evidence from morphology, comparative anatomy, looking at what they look like and comparing one group to another to decide what belongs here, and here. But problems do arise. Let's take a look at another example. In this example here, we look at the Phylum Chordates. Just this section here of the Phylum Chordates, with the land animals that belong to the Phylum Chordates, and some ancestral chordate, a simple organism again from the ocean, like I mentioned previously. Amphioxus, the lancelet, is a very simple chordate that has that dorsal structure called the notochord in it, and all of these do have that similar structure which is related to the backbone. So all of these belong to the phylum Chordata. But if we laid out all of these classes, again in the form of this cladogram, using all of the groups that we studied in 5.3, amphibians, then reptiles, then birds and mammals, each evolving in time much later in the fossil record than we've had reptiles and amphibians. And you might think that's totally correct. But evidence from gene sequencing and molecular cladistics has proven something that allows us to now describe reptiles. When you look at what belongs inside of the reptilia, and let's take the reptiles out of this diagram now. Here in this cladogram, what we see is that there's an ancestor here, and that ancestor gives rise to the group that we call the reptiles, including things like turtles, snakes, lizards. But there's another line of evolution that gives rise to crocodiles and then to birds. What this is showing is that crocodiles actually don't belong with this group, the reptiles, and they should actually be placed in some kind of group with the birds. So if you click on the card above this video, it will take you to another video that I made some time ago talking about all of this in more detail. So I want you to go have a look at that to understand why there's a need, of course, to move away from the traditional classification system, to look at a completely new way of laying out the tree of life, which is based on what we call molecular cladistics or molecular phylogeny. You can also click on this other card here, which is a lecture by a Yale professor talking about some very interesting anomalies or exceptions to what we've had before. And he mentions this example, of course, about crocodiles and birds in his lecture. And that is actually where it was drawn to my attention first. So you can go and watch the first 15 minutes or so of that lecture if you're up for that. Now for this week's main activity. Because we've realized that there is some reason to want to reclassify the tree of life, then we have to look at the skills that we need to understand how to produce these molecular cladograms. And this is what we are going to look at. Using the power of modern databases, we are going to go in and interrogate or get data from databases, get some gene sequences, put together a few tables, and get some data so that we can understand how cladograms are constructed, so that we can tell whether a chimpanzee is more related to a human being, or whether a gorilla is more related to a human being, or whether an orangutan is more related to a human being. We are going to go in and look at how to acquire this data, how to organize it, and how to construct our own simple cladogram for the primates. So let's move in and take a closer look.